gluconeogenesis and glycolysis. Although this process shares the same enzymes as glycolysis, it is not the reverse process. It is more complex because there are more steps involving enzymes outside of the glycolysis process. Gluconeogenesis is the production of glucose from non-sugar precursors. It mainly occurs in the liver and involves the synthesis of glucose from compounds that are not carbohydrates. When the cell is growing on the other carbon compounds, glucose must be synthesized. For most of the steps of gluconeogenesis, the same enzymes are used like glycolysis. In step 7, fructose 1,6-biphosphate to fructose 6-phosphate, and in step 9, glucose 6-phosphate to glucose are the unique ones. Both of these steps require different enzymes than used in glycolysis. This is because in glycolysis, these steps actually require energy to occur, so the same enzyme isn't needed when doing the reverse. In our body, the brain can only use energy that has come from glucose, yet the body can only store enough glucose to last for less than 2 hours. It's through gluconeogenesis, which is a pathway used by the body to create glucose from other non-carbohydrate molecules. This is the pathway that prevents us from dying when we haven't had any bread, vegetables, or other carbs for a few hours. For example, if a human strictly ate meat all day and nothing else, no carbs, no vegetables, nothing. Although the pathway starts with glucose, the majority of sugar that we take in is not glucose. Apart from starches, humans take in a large amount of sugar as sucrose, so table sugar, lactose from milk, and fructose from corn syrup. We're going to break down each of these steps and figure out how they build starting on the map. So sucrose is composed of glucose and fructose. Lactose is composed of galactose and glucose, as you can see in the beginning of my poster in these pictures below. Sucrose can be broken down into glucose and fructose by the enzyme sucrase. This gives us direct entry for one of those sugars. From here, I investigate how fructose is turned into glycolysis metabolite. There are 2 different pathways that these processes can occur. The process also covers how the body metabolizes fructose as is a metabolite in sucrose breakdown. This leaves us with lactose, which is broken down by the enzyme lactase into glucose and galactose. So for Lab 5, this image below shows the worksheets from Lab 4, and then I added on to Lab 5. So I'm going to do a little recap. So this lab focused on key mechanisms within the citric acid cycle and bridging that to the glycolysis pathway. In this lab, I dissected the citric acid cycle. I determined how molecules from electron transport chain can be turned into energy for the cell, examined the components needed for cellular respiration, and made a diagram of the steps of the pyruvate dehydrogenase complex. There is a gap between pyruvate and acetyl-CoA. This is accomplished by the pyruvate dehydrogenase complex. This process of transforming pyruvate and acetyl-CoA requires the use of 5 coenzymes. These include thiamine pyrophosphate, flavin adenine dinucleotide, nicotine adenine dinucleotide, coenzyme, and lipoamide. This process of transforming pyruvate and acetyl-CoA requires the use of 3 enzymes. These enzymes are TPP uses pyruvate dehydrogenase, FAD uses dihydrolipoyl dehydrogenase, and lipoamide use dihydrolipoyl transacylase. I used a diagram of a mechanism of the pyruvate dehydrogenase complex. I included all the enzyme cofactors and structural intermediates, as you can see in worksheet 12. Then I investigated different amino acids that can be made from the citric acid cycle. Then I investigated aspartate and glutamate both can be formed from intermediates in the citric acid cycle. To make them, we use an enzyme called aminotransferase. This enzyme will take a carbonyl group and exchange it with amine. I made a diagram that shows the process of generating aspartate and glutamate in worksheet 13. Alanine can also be made by the same process. Then I made a diagram and used structures, and I labeled the precursor, as you can see in worksheet 13. Now, as we move on to the electron transport chain, it is composed of 6 enzymes used to transport. These 6 enzymes include NADH dehydrogenase, succinate dehydrogenase, cytochrome C oxidorreductase, cytochrome oxidase, glycerol 3-phosphate dehydrogenase, and acyl coenzyme dehydrogenase. The electron transport chain also relies on 3 coenzymes. The 3 coenzymes and which enzymes they are linked to is NADH dehydrogenase is FMN, succinate dehydrogenase is FAD, cytochrome C oxidoreductase is HEME, FES, cytochrome oxidase is HEME, and acyl coenzyme dehydrogenase is CoA. I also did a diagram of the process and electron transport chain in worksheet 11, And then I took a look at protons that make ATP. So ATP synthase is composed of 2 main regions. The F0 region, which is essentially a long tube for transporting, and the F1 region, where the ATP is made. The 5 polypeptide chains that make up the F1 subunit of the ATP synthase is alpha, beta, gamma, delta, epsilon. Rotation, the ATP synthesis mainly use 3 of the subunits, the A3, B3, and Y subunit. In worksheet 6, I made a diagram of the 4-step process generating ATP from ADP. Then I made a diagram of the process of the proton pump. So here's lab 6. This includes the worksheets from lab 4 and lab 5, and then I'm just slowly building up my poster all together. So for this lab, I focused on key mechanisms of amino acid generation and bridging them to the citric acid cycle and glycolysis pathway. I examined the pentose phosphate pathway and made a diagram for the processes of fatty acid degradation. The pentose phosphate pathway generates some sugars that I've already seen in the previous labs. The pathway will begin with glucose 6-phosphate, the major metabolic product of glycogen degradation, which the 2 sugars from the pentose phosphate pathway used in glycolysis is fructose 6-phosphate and glyceraldehyde 3-phosphate. Another sugar emerges from this process. One that is used in DNA synthesis is ribose 5-phosphate. The pentose phosphate generates the reducing molecule NADPH. 3 enzymes that use this reducing molecule and the reaction it performs is glutamate dehydrogenase is involved in reductive amination. Glutathione reductase is involved in protection against ROS, and NADPH is also used for cholesterol biosynthesis and fatty acid chain elongation. Every step in the breakdown of glycerol can be used in a different metabolic pathway. Although we have yet to learn, at this point, we haven't learned about the L-glycerol 3-phosphate, but the 2 metabolic products are in the map that you can see below. 2 end products of glycerol degradation and all the possible places within the metabolism that these molecules could go. So the first one is fatty acids produced are oxidized by beta oxidation into acetyl CoA, which in turn is used in the Krebs cycle or the citric acid cycle. Glycerol released from triglycerides enters the glycolysis pathway. In order to break down the fatty acids, we have to generate the acetyl coenzyme. 2 additional reagents needed for this transformation is ATP and the coenzyme. Fatty acid degradation is a slow process that occurs by cutting out 2 carbon units at a time. All the reagents that are needed in this process for the fatty acids is acyl coenzyme dehydrogenase to form enol coenzyme hydratase to form 3-hydroxylacyl CoA, hydroxyacyl CoA dehydrogenase to form beta-ketoacyl CoA, and thiolase to form acetyl CoA. The purpose of FAT in terms of mechanism is that it is a redox-active coenzyme associated with proteins. It is involved in various enzymatic reactions in our metabolism. The purpose of H2O in terms of mechanism, so H2O in terms is added to the second step with the help of the enol coenzyme hydratase. Hydratase so as to convert the alkaline bond between alpha and beta acyl enol coenzyme into beta hydroxyl acyl coenzyme. The purpose of CoA in a mechanism is known for its role in the synthesis and oxidation of fatty acids and the oxidation of pyruvate in the citric acid cycle. Acetyl-CoA can be transformed into water-soluble ketone body. The advantage of this over fatty acids is that ketone bodies can cross the blood-brain barrier and therefore available as fuel for the cells of the central nervous system, acting as a substitute for glucose, in which these cells typically will survive off of. So here is my poster complete altogether with all 15 worksheets, and now I'm going to do a recap of Lab 7. So in Lab 7, I learned about the nucleic acid generation and bridging them to the citric acid cycle and glycolysis pathway. I dissected the process of lipid, cholesterol, and amino acid synthesis and found the difference between how different types of lipids can be broken down, saturated versus trans, and unsaturated. I used worksheet 5 to diagram the process for transforming uridylate into UTP, CTP, and TTP. I also used worksheet 5 to show the process for transforming inosinate into ATP and GTP. From the cholesterol pathway, there are 36 ATP required to generate this compound. A reducing agent is used within the process that we previously generated. The reducing agent where this is synthesized is NADPH that belongs from the pool of this compound in the cytosol. I used worksheet 15 to diagram the process of generating cysteine from methionine. A side product of this process is a metabolite from the citric acid cycle. The side product produced in the acetyl-CoA, which is used in the citric acid cycle. It fits in the citric acid cycle in the very first step. So in acetyl-CoA, oxaloacetate forms citrate using the enzyme citrate synthase. I used worksheet 10 to diagram the process generating serine and then glycine from 3-phosphoglycerate. The source of the 3-phosphoglycerate is serine. I also used worksheet 10 to diagram the process of generating arginine from glutamate and acetyl-CoA. The essential amino acid in tyrosine is made from phenylalanine. So that is my complete poster and my recap of Lab 4 through 7. I know it was quite lengthy. So nucleotides can be synthesized by a variety of means in both vitro and in vivo. In vitro, protecting groups may be used during laboratory production of nucleotides. A purified nucleoside is protected to create a phosphoramidite, which can then be used to obtain analogs not found in the nature to synthesize an oligonucleotide. In vivo, nucleotides can be synthesized de novo or recycled through the salvage pathways. The components used in the de novo nucleotide synthesis are derived from biosynthetic precursors of carbohydrate and amino acid metabolism, and from ammonia and carbon dioxide. The liver is a major organ of the de novo synthesis in all 4 nucleotides. I did mention that in one of the previous labs. De novo synthesis of pyrimidines and purines follow 2 different pathways. Pyrimidines are synthesized first from aspartate and carbamyl phosphate in the cytoplasm to the column precursor ring structure orotic acid onto the phosphorylated ribosyl unit is covalently linked. Purines, however, are synthesized from the sugar template onto which the ring synthesis occurs. For reference, the synthesis of purine and pyridamine nucleotides are carried out by several enzymes in the cytoplasm of the cell, not within a specific organelle. Nucleotides undergo breakdown such as useful parts can be reused in the synthesis reaction to create a new nucleotide. Ribose is a type of sugar normally made in the body from glucose. Ribose plays important roles in the synthesis of RNA, DNA, and ATP. The pentose phosphate pathway is a metabolic pathway parallel to glycolysis. It generates NADPH and pentose, as well as 5-phosphate, the last one a precursor for the synthesis of nucleotides. Glycolysis begins glucose metabolism in all cells to produce 2 molecules of pyruvate. Occurs outside of the mitochondria. It's usually in the cytoplasm. Cellular respiration uses oxygen from the environment and converts each pyruvate to 3 molecules of carbon dioxide while releasing ATP. And finally, ATP is a nucleotide that consists of 3 main structures: the nitrogenous base, adenine, the sugar ribose, and a chain of 3 phosphate groups bound to ribose. The phosphate tail of ATP is the actual power source which the cell taps. ATP synthase is an enzyme that creates the energy storage molecule ATP. ATP is the most commonly used energy currency of cells for all organisms. ATP is a nucleoside triphosphate where a high-energy bond attaches to a third phosphate group to the molecule. The bond is highly unstable, and when it is hydrolyzed, it releases a substantial amount of free energy. In addition to providing energy, ATP has other essential roles within cells.